So thank you so much for giving me the opportunity to talk to you today about all the tools that Takara Bio has developed for the gene editing and culture of iPSCs. So this is a first glimpse of the agenda. First, I'm just going to talk a little bit about the key considerations that you should think about when you are designing your gene editing experiment. And then we will follow with a test case when I'm going to explain to you how we edited hiPSC cells to introduce a SNP related to tyrosinemia, followed up by differentiation to hepatocytes of the edited clonal cell line. And then a brief summary. So this, as you can see here in the gene editing workflow. So first, from your hiPSCs, we edit the cells introducing the Cas9 and the HDR template, usually as we do at Takara Bio by electroporation. Then at the end, you will have an overall edited population. Some of the cells will be wild type, some other carry the SNP of interest. And then we will move forward towards the isolation of these single cells, then expansion of these clones. And then one of the key points is actually screening these clones in order to find the clone that has been successfully edited, followed by an expansion of this edited clone. So here are the key considerations when you are trying to design your editing experiment. So the first step is actually design the guide RNA and the repair template. And the first thing that I always say to people is that you really need to sequence the target site around your the sequence around your target site on your edited cell lines, just because many times there are some SNPs that can actually affect the sequences of possible guide RNAs that are in this region. Then you need to pick single guide RNAs as close as possible to the modification. This is especially important if you are introducing a SNP. You usually need to have the cut site of the single guide RNA 10 bases upstream or downstream. If you pick any guide RNA further away, then the efficiency of your HDR will actually decrease. Another good practice is actually check the activity of the single guide RNAs in vitro. This actually first step can really rule out some guides that due to their sequence, they are not very active. Then another good thing is just to use an optimized single guide RNA scaffold. We actually have an in vitro transcription kit that allows you to produce a single guide RNA that has certain modifications in the scaffold, like actually an extra loop that will increases the complex of the Cas9, making it more stable. Then a good practice, and especially if you are working with an hiPSCs, is actually use single-stranded donor templates because they have lower rate of random integration if you compare it with a double-stranded DNA, and also have a lower toxicity. It's important as well, and as I mentioned yesterday, to in the template oligo or in the HDR template to mutate the PAM if it's possible. This actually will prevent the recut of the Cas RNA of the Cas9 to the target site if the editing has been successful. Or if it's not possible to mutate the PAM, just introduce silent mutations in the seeding region of the single guide RNA that is actually 7 to 10 base pairs upstream of your PAM. And if you are actually knocking SNPs, it has also been shown that the template polarity, meaning if you are using the sense or the antisense strand, has an influence on the HDR efficiency. Usually the rule is that if your double strand break is upstream of your insertion site, you should actually use the antisense strand. And if it's downstream, you should use the sense strand. When you are moving towards the introduction of the Cas9 and the guide RNA and the template repair into cells, one of the key points is actually minimize the cell toxicity due to the delivery of this complex. And one of the best practices is actually doing it on the form of the RMP complex, meaning the protein of Cas9 complex already with the guide RNA. This actually has the lowest toxicity in cells. You don't need to full transcription or translation machinery, and actually because there is a relative low transience, meaning that the cell, usually the Cas9 is between 24 to 48 hours inside the cells, there are less of target effects. And there are no integration events that could happen, for example, if you are introducing the Cas and the single guide RNA in the form of a plasmid, encoding in a plasmid. Then one of the key points of this type of experiments is actually the single cell cloning if you are working with hiPSCs, because isolating and then trying to expand single cell clones is actually very challenging with iPSCs because a lot of them, they die or they actually differentiate. So therefore, it's very important to have a media system that allows that. And at Takara Bio, as Malathi mentioned, we have developed the DefCS, that is a cell culture system that allows the culturing of iPSCs in a monolayer, and it's feeder-free. And actually, one of the great characteristics of the DefCS is that enables the survival and the expansion of the single cells. That's actually key in any editing experiment. And at the same time, even allows the survival of the single clones, maintaining the undifferentiated state. So therefore, when you are expanding, you can differentiate the edited clones that you desire and maintains a normal karyotype. And the final, when actually you have single clone, isolate the cells, and you have expanded them, then it comes the screening and the validation. And actually the screening can be a very daunting task, because many times you will have a lot of 96 well plates that you really need to screen fast, because your cells are growing actually in different rates inside the wells. So there is the need of a fast and reliable method to detect the specific edit. And actually, this can be challenging if you are actually detecting only the introduction of a SNP. And that's why at Takara Bio, we develop what a guided knock-in screening kit. That it's a kit that allows you to detect the specific edits on the clonal cell populations. So I will just follow through about how is the workflow of this kit. So imagine you want to introduce a G to an A mutation. So after the edit, single cell clone isolation and expansion, we will copy the plate. And then from a 96 well plate, we will just extract the genomic DNA and PCR amplify the region of interest. This case here. Then this PCR product is actually hybridized with two probes. These probes are actually standard oligos that you can order in any synthetic company, and they are depending on your sequence. Actually, at Takara Bio, we have an online tool that allows you to design these probes just only by the introduction of your target sequence. So one of these probes is called displacement oligo, hybridizes with a specific TM 5' of your specific SNP and has an extra base on the 3'. Another probe, it's called flap probe oligo and contains actually two sequences. One in the 5' is a fixed sequence. We actually provide this sequence to you. And then it actually is encoding for the SNP of sequence and hybridizes on the 3'. So after a full hybridization, you will have what it's called a double flap structure. This structure is actually recognized by this enzyme that we call guided flapase. So after this structure, the guided flapase will cleave this fixed sequence, and the release of this fixed sequence will trigger a fluorescence signal. If instead of the SNP, you still have the wild type sequence, you are generating, as you can see here, what we call a gap structure. So this structure is not recognized by the guided flapase, and therefore there will be no fluorescence detected. So actually, this will allow you that this fluorescence, I forgot to say, it actually can be detected by a plate reader. So you will introduce your plate reader, you will have a fluorescence readout, and whatever is positive, it will mean that it contains your desired SNP. We also have a second version actually of this kit that allows not only the generation of one fluorescence, it's also another a red fluorescence. So therefore it can also be applied to the screening of heterozygous clones, having the SNP being green, for example, fluorescence, and the wild type being red. And the overall of the workflow is around 4 hours. So I'm just now changing gears and showing you how we have applied all these tools in the specific case of introducing a SNP related to tyrosinemia. So tyrosinemia is a disease related to a mistake of the metabolism, because in the body they cannot really effectively break down the tyrosine. One of the types of tyrosinemia is called type 1 and is related to the mutations on this FAH gene that encodes this enzyme fumaryl acetyl acetate, that it's one of the enzymes that breaks down in multiple steps the tyrosine. So we just really wanted to generate hepatocytes that they were carrying this SNP. That it's a G to an A, so it mutates a tryptophan to a terminated step codon. So following my advice before, you just look here in red is the SNP, the base that we want to mutate to an A. And then you have here actually two guide RNAs that are 10 bases upstream of the SNP that we want to introduce. And actually, the mutation of a G to an A will cause the PAM to be mutated. So actually, we don't need to introduce silent mutations here. It's already, the SNP will itself mutate the PAM. So, and in this case, we in vitro transcribed the guide, and then we electroporated independently hiPSCs with guide number 1 and guide number 2. So many times you also will face that you will have guides that their in vitro were active, and then once you are edited, say how I really detect that the knock-in has happened, and which guide do I know has given me the highest percentage of knock-in. And in this point is actually when you can actually apply the guided knock-in screening kit. It not only detects the mutations on the clonal cell lines, it also can be applied on the bulk edited population. So in this case, actually you can see how guide RNA number 1 give us a higher fluorescence level, and therefore there is more percentage of successful knock-ins in this population than in the guide RNA number 2. So the next step was actually single clone isolate this population, and here you can see a timeline of all the different steps that I mentioned before. In this case, actually, we single clone isolate, performing limiting dilution. And in this case, we calculate a dilution that will give us 50 cells per well. And you can see here we have 596 well plates. And in total, at the end, we had a survival rate of around 93%. We also have performed some other editing experiments that we single cell isolate instead of limiting dilution by flow cytometry. And in this case, the survival is less because the whole process is more harsh for the cells because they go through the sorter. And usually in that case, the survival rate is between 60 to 70%. So after the single cell isolation, we expanded them and we applied the guided knock-in screening kit, as I mentioned before, to be able to detect which clones they really have or G to an A mutation. So here is actually the usual readout. You will have the clones, and then when you go through the plate reader, you will have a baseline signal. This is the background signal. And then some of the clones, we also have a very nice, intense green signal. These ones actually were the ones who have the A instead of a G in that specific gene tyrosinemia. So then in blue here are marked the ones who were homozygous, and in orange they are the ones who are heterozygous. So we took the homozygous clones, we actually rechecked the signal of the guiding knock-in screening kit by Sanger sequencing, as well as we check the pluripotency of all of them with high levels by flow cytometry measuring OCT4, TRA160, and SACF4. So as the next step was actually to take this clone 181, and then we perform the differentiation to hepatocytes. So in order to perform this differentiation, we use a product that allows the hepatocyte direct differentiation from hiPSCs. And it contains actually in two phases. The first one, it's the differentiation to the definitive endoderm, and then the second one is to the hepatocytes. So it's a standardized serum and feeder-free protocol and recapitulates the liver development in vivo. It has all these different steps, and you can see how each of them has a specific morphology. And at the end, it yields a poor population of hepatocytes. So in this case, we perform the differentiation of the clone that I mentioned about before, the 181. By microscopy, you can see a very nice morphology and pretty homogeneous with a polygonal shape. And then we also wanted to ensure the complete differentiation. And for this reason, we perform a staining and quantification of this transcription factor, HNF4 alpha, that it's actually a successful transcription factor for differentiation of hepatocytes. And we found that actually more than 92% of our cells were positive. And then as a functional test, one of the key points of any hepatocytes is the drug metabolism. And it's specifically the detection of the activity of certain enzymes that metabolize drugs, specifically the family on the cytochrome 450 or actually CYP family. So this type of assays, the readout is done by LCMS, and you can see the degradation of all these different drugs performed by different enzymes of this family with similar levels between our control and our edited line. So with all these results I have shown you, how we can perform an successful edit, and then these edited cells can be differentiated, in this case with the hepatocytes. So as a summary, the take-home message is that the gene editing requires a high survival and maintenance of pluripotency, and a successful culture and clonal expansion is actually key. And in Takara Bio, have developed a range of tools that will help you to edit the cells and also differentiate them in hepatocytes. And that's all, and thank you, and I will take any questions.